Hey BioKids, this is Mr. Wolf, and in this video we'll be talking about incomplete dominance and codominance and how codominance applies to human blood typing problems. Let's first talk about Mendel's law of dominance. Now, Mendel stated that there's always going to be a dominant trait masking or covering up a recessive trait. In the case of his pea plants, purple flower color always masked white flower color. And so in the heterozygous condition, we always had purple flowered pea plants. So if we had two pure pea plants that were bred, they would always result in having heterozygous purple flowered offspring. Now, this is not the only type of dominance we see in genetics. Another example of inheritance is codominance. Now, in codominance, both traits are going to be expressed equally. In this example, we have feather color in chickens. Both black and white feathers would result in the heterozygous condition. Now I use special notations for this. I use a capital F to represent feather colors, and I use capital B for black feathers and capital W for white feathers. Notice they're both capitalized because they're both dominant. They don't cover up each other. Both will be expressed equally. So if we cross a purebred chicken with black feathers and a purebred chicken with white feathers, they'll produce an offspring that have both black and white feathers. It looks something like this. There'd be my black feathered pure dominant chicken. There'd be my white feathered pure dominant chicken. When we cross them in a Punnett square, all the offspring would be heterozygous for feather color, but that would result in a black and white feathered chicken. A different type of dominance inheritance is incomplete dominance. In this case, we'll use just capital letters. We're not going to use superscripts as we did in codominance. Now in codominance, both alleles expressed equally, but in incomplete dominance, we get a new third phenotype that appears in the heterozygous condition. And oftentimes it's like a blending of the two traits. In this example, we use red and white flowers. These happen to be snapdragon flowers. If we get a heterozygous red and white genotype, a big R, big W, this results in pink flowers. So if we cross a purebred red flower with a purebred white flower, all their offspring would have pink flowers, even though they have two different alleles for that gene. There's no pink flower gene, it's that the mixing of the red and the white gene produces pink flowers. Now at this point in the video, I'd like you to stop and take a look at the incomplete dominance and codominance problems I've assigned for you. Once you've completed those, you can move on. Now the next part of this video is setting up blood typing genetics problems. These tend to be the most difficult problems we do in class. So this step-by-step walkthrough will help you set up these very challenging dihybrid problems. So first of all, these are going to be dihybrid crosses, which means we'll be following two different traits in this example. So we're going to cross a woman who is heterozygous for AB negative blood and a man who is heterozygous for A positive blood. Now you've probably heard of blood typing before. Maybe you know your blood type. For example, my blood type is O positive. But what does that really mean? Well, let's go over our symbols. We'll go over our parent phenotypes, our parent genotypes, just like every other problem we've done. So our first symbol is going to be capital I, A. And notice we use a superscript because this is going to be a codominant allele. And what this gene or this allele codes for is a type A antigen or a glycoprotein that is found on the red blood cell's surface. So in our red blood cells, we have all these different kinds of proteins and sugars that coat the surface that act like different chemical messengers and different chemical markers. And this happens to be the A protein. It's actually a glycoprotein. So if you have type A blood, that means all your red blood cells has this little type A antigen on its red blood cell surface. There's also a type B antigen, which is actually a mutation of the type A antigen. But this type B antigen is codominant, so we use a capital I, capital B to represent that type of allele. And these proteins are just slightly different, or these glycoproteins look a little bit different than the A proteins, so they're slightly different because they're coded by a different gene. Now, if you inherit both a type A and type B antigen allele, you will have what's called type AB blood. That's how we get type AB blood people in this world, because both the A and the B antigens are expressed equally, and your red blood cells have both types of glycoproteins on their surface. Now there's a third allele, a third mutation occurred for this type A or type B antigen, and this actually codes for no antigen, which gives us type O. Notice we use a little I for this because this is recessive, which means if you have a type A and a little I antigen, or if you have a type A and a little I allele, you will produce type A antigens and then also no antigens, so the A kind of overrides the no antigen gene. The only way you can get type O blood is if you have two little i's, two recessive alleles, as if it was any other recessive trait. Notice the type O red blood cell has no antigens on its surface because there's no protein or glycoproteins being coded for. So that covers the ABO part of your blood type. Now, if you notice, there's also a negative positive part of your blood type. Now, I promise you, this is a little bit easier. This stands for the Rh factor. You can be either Rh positive, which is dominant, or Rh negative, which is recessive. So we're following straight up dominant recessive Mendelian law of dominance traits. The reason why it's called the Rh factor is actually because it was discovered on rhesus monkey blood first, and we found that we also have this blood protein on our red blood cells as well. So now that we have all our symbols written down, what we'll do first is write down the parent phenotypes. So our mother is AB negative, and our father is A positive. Now, figuring out the parent genotypes would be pretty easy because we have those descriptive words like heterozygous written into the problem. So the AB negative mother is capital IA, capital IB. In fact, that's the only way you can be type AB blood. And she's also negative for her Rh factor, which means she has to have 2 little r's. That's the only way you could be Rh negative. Now, the father is heterozygous for A positive. We're going to assume that the heterozygous applies to both the A part of his blood type and the positive part of his blood type. So for his type A part, he is capital IA, little I. He's heterozygous, so he only has 1 gene, a glycoprotein, and he's big R, little r, because he only has 1 gene for the Rh factor. So there are our genotypes. So that means the mother has the AB negative blood, which looks like this, and the father has the A positive blood. Notice that extra glycoprotein orange on his red blood cells. So these are what the blood cells will actually look like. So now let's figure out our possible gametes. Fortunately, this is the same process we've used before in other dihybrid crosses. We're using the FOIL method to figure out all the possible combinations from both mom and dad on what type of genes they could pass on to their children. So let's take a look at mom first. If we look at the first I and the first R, she would give a capital IA, little r for her first possible combination. Now, if we go to the outer ones, notice it's the same, capital IA, little r, so we don't need to write it down again. The inner letters would be capital IB, little r, so another possible combination, and the last letters, capital IB, little r again, so no need to write it down. So she has 2 possible gamete combinations from her genotype. Now the father's actually going to have 4 because he's heterozygous for both. IA, big R for first, IA, little r for outer, little I, big R for inner. Notice I keep the letter order consistent even though the I is little. I keep it before the R. And then little I, little r for the last possible combination for his gametes. So the father has 4 possible combinations. So this sets up our Punnett square nicely. We're going to have a 2 by 4 Punnett square. So this 2 by 4 Punnett square we set up like this. I prefer 2 rows and 4 columns. You can set up the other way if you want to. But we take the 2 combinations, put them on the side here, and then the 4 possible combinations from dad and put them up top, and we just fill out the Punnett square, keeping the I's in front and the R's following. So IA for the first box, big R, little r. IA for the second box, little r, little r. And we just go through keeping the I's in front of the R's, and we fill out all of our possible combinations. So these are all the possible combinations of children that this set of parents could have. Now we just need to figure out the phenotypes of these possible kids. This first one is going to be A positive blood. Next one, A negative. Another A positive. Another A negative. AB positive. Notice we have an IA and an IB, which means it's AB blood. It's codominant. AB negative, B positive, and B negative blood. Notice the only type of child they can't have is a type O child. And we'll find this all the time when we have an AB parent. Whenever there's an AB parent, there is no possible way they could have a type O child because they don't have a little I to donate to their children. So that's blood typing. It's pretty complicated, but once you get past that gamete part, it's pretty straightforward. Feel free to watch this video again if you're having trouble. See you in class. If you're interested in learning the importance of blood typing, check out Mr. Anderson's video that I have linked above me. Just click on the lowercase I.